Hey everyone, and welcome to the first episode of Reef Aquarium Science. The aim of this series is to try and translate scientific research into practical knowledge that aquarists such as myself and you guys can utilise in a way that's useful at the hobby level. In this video, I'll first dissect several relevant scientific papers on light, nutrients and water flow and discuss how the implications of these findings can impact our hobby. So the first study comes from Standish et al. 2023, and this one examines how long-term nutrient regimes influence coral physiology. The species selected was Acropora polystoma, a branching acropora with high metabolic demands and fast skeletal deposition, and Priorities lichen, a more slow-growing massive coral that allocates resources differently and is often more conservative. Both were cultured for 140 days under 4 different nutrient regimes. The balanced regime maintained approximately 0.28 ppm of nitrate and about 0.057 ppm of phosphate. The depleted regime had basically 0 of both nutrients, and the nitrate-only regime held nitrate around 4.5 ppm and no phosphate. The phosphate-only regime supplied roughly 0.5 ppm with no nitrate. So across both these studies, balanced conditions maintained steady calcification and tissue condition. However, under depletion, skeletal growth slowed, tissues thinned and polyp extension reduced, and colonies became more sensitive to light exposure that would otherwise be tolerable. Under nitrate-only conditions, the symbiont densities increased and the colonies darkened, but the absence of phosphorus constrained ATP production and membrane turnover caused skeletal growth to not keep up, and the colonies remained physiologically brittle. Under phosphate-only conditions, the bottleneck flipped, so proteins could not be synthesized at normal rates, tissue maintenance faltered, and colonies reacted poorly to routine handling and light changes. A common thread was that imbalance or depletion increased the likelihood that ordinary lighting became a stressor rather than a driver of growth, which is what we find in our hobby sometimes. These results align closely with what many aquarists have observed without necessarily having the mechanistic explanation. So systems that drive nitrate and phosphate to undetectable levels often produce pale, brittle SPS that fail to extend polyps. At the other extreme, systems that carry several parts per million of nitrate while running phosphate near 0 often show darker corals with apparently fuller tissue, yet growth edges stop and bases recede when conditions fluctuate. Nitrogen is a backbone for amino acids and enzymes that run photosynthesis and calcification. Phosphorus is also essential for ATP and for phospholipids that also form membranes. Remove either of these and the symbiosis cannot process light efficiently. The excess energy has nowhere productive to go, basically. In terms of real-world applications in our tanks, the first conclusion is to avoid chasing true 0 for both nitrate and phosphate. Test kit 0 is not really an achievement if it coincides with suppressed growth and increased stress. The second conclusion is to avoid skewing the system by removing one nutrient selectively or by dosing one without the other. Running phosphate removal media to absolute depletion while feeding heavily or dosing nitrate leaves the symbionts unable to make the ATP necessary to handle light safely. So dosing phosphate without nitrogen leaves the animal short of proteins needed to build the tissue. It is far easier to keep Acropora growing under moderate, stable nutrients and to try and force them to adapt to oscillations and extremes. The second study by Han et al. 2022 tested how small balanced nutrient conditions influence bleaching under combined heat and light stress. The team used 3 Hawaiian species that represent different life history strategies. So they use Libactis scutaria, if I can pronounce that correctly, a free-living large polyp coral, Montipora capitata, which is a common one that a lot of us have, like I have it in this tank here, and Posilopora acuta, a fast-growing branching coral with high light demand. The experimental temperature target was about 30.5 degrees Celsius, and light levels were set to high to ensure the photosystems were being pushed. This is a combination that triggers bleaching in the field and in marine aquarium settings. Half the treatments received no added nutrients, the other half received very small balanced additions of nitrogen and phosphorus, with less than 0.1 ppm, which is well below the detection thresholds of many test kits that us hobbyists use. The difference in outcomes was significant, so the corals without added nutrients bleached quickly, symbiont densities fell, pigments were lost, and calcification slowed or stopped altogether. Corals with balanced additions lost colour more slowly. In Montipora and Posilopora, the calcification persisted at higher rates despite thermal stress. However, Labactus was less responsive, which is a reminder that different species will also behave differently to different stresses. So in this situation, heat and high light damage the protein complexes that capture light, and repairing these requires different amino acids and energy as well. Nitrogen provides material for new proteins, and phosphorus is vital to ATP, which powers these repair steps. When both are available in small amounts, repair keeps going for longer. However, when either is missing, damage outstrips repair and bleaching accelerates. For Aquarius, this means that a tank operated at absolute zero nitrate and phosphate under strong LEDs is basically going to bleach when temperature or light increases, whereas a tank holding a small balanced baseline is a bit more resilient. And I think it's important to talk about here when we're talking about zero nutrient levels and stuff like that. Obviously there's a lot of other stuff in the water, but we're talking about just pure nitrogen or phosphate in the system. If you're feeding really, really heavy each day and still getting low nutrients, you've probably got those elements in the system, but it's just being stripped out. But we'll talk about that later, how it's important to continuously and consistently dose things in the tank rather than just hitting everything at one time. So our third paper is work in 2013 by Wiedemann et al. And this showed that excess nitrogen with limiting phosphorus increases bleaching risk. Many aquarists have created exactly that scenario unintentionally by dosing nitrate or feeding heavily whilst running aggressive phosphate removal. Colonies become visually darker and symbiont densities rise, but the shortfall in phosphorus means that the light absorbed cannot be processed safely when conditions change. The system appears stable until a minor heating event or a slight increase in PAR or a change in flow shifts the balance and the corals will decline quickly. So essentially, running no phosphate isn't good either, because it means that whilst your corals might look all right, any sort of little change you get is just going to make them die, basically. So the next paper by Becker et al. Examined what happens over long timescales when corals receive a constant low-level enrichment of both nitrogen and phosphorus. The study location was a foreshore reef in a oligotrophic environment where background nutrients are naturally low. The enrichment was low but continuous for 15 months. Posilopora was used as it's a fast-growing branching coral and often used as an indicator species for the reef condition, often because it responds quite quickly to change. After more than a year, colonies in the enriched plots looked and functioned quite differently. Symbiont densities were about 54% higher than in control plots, and tissue biomass increased by about 27%. So the maximum photosynthetic rates were also about 29% higher, and oxygen production under moderate temperature was roughly 30% higher as well. Essentially, that means that the colonies had thicker tissues, more photosynthetic capacity, and larger energy reserves to draw upon when conditions changed. This is relevant to us reef keepers, as corals not only react to current water parameters, but adapt to them over months. A tank that runs zero nitrate and phosphate may display bright colours in the short term, but the colonies lack nutritional reserves. They do not have the tissue depth or symbiont populations to withstand any sort of instability or change. By contrast, a tank that measures a very low but consistent baseline of both nutrients allows colonies to build capacity. They thicken tissue, expand symbiont populations, and increase photosynthetic output. Those reserves translate into tolerance when PAR is increased or when seasonal temperature rises occur out in the natural environment. The fourth study by Lentz et al. In 2024 investigated the role of flow in growth and survival. So the species tested include Montipora capitata again, Posilopora acuta, and Parietes compressor. Flow was shown both as tank turnovers per hour as well as velocity in centimetres per second. Growth increased as flow rose from stagnant to moderate levels, approximately 10 to 30 centimetres per second. At those rates, colonies built tissue and skeleton efficiently. When flow exceeded roughly 50 centimetres per second, growth then declined again. So Montipora capitata doubled growth between 0 flow and 105 turnovers per hour, then fell back at 210 turnovers. Posilopora acuta performed best at 30 and declined under the highest flow. Survival during and after bleaching followed a similar pattern. At moderate flow, survival ranged from roughly 75 to 100%, depending on species. At very high flow, survival dropped towards 25 to 58% range. The shape of the response curve is not really surprising when considering alongside the physics at the colony surface. Moderate varying flow collapses the diffusive boundary layer across the top of the coral, and it also carries away any reactive oxygen species generated through photosynthesis or any other sort of waste products. Keeping local chemistry within tolerable bounds, basically. So insufficient flow allows those byproducts to also accumulate. An excessive unidirectional flow imposes mechanical stress and metabolic costs, basically. So when applied to our system, this means flow settings should be planned and revisited whenever lighting or feeding schedules change. 2 or more pumps positioned to create alternating crossing streams will produce reversing field, which reduces stagnation at the colony. It also sweeps waste away more efficiently than a single consistent jet, even if the nominal flow rate's the same. So basically, like if you've got 1,000 litres per hour going in one direction, that's going to be nowhere as good as if you split that between 2 different pumps, still had 1,000 litres per minute or per hour, whatever it was, across both pumps, but they were oscillating like that, so your water's going backwards and forwards. You'll probably be able to see in the tank behind me that that's kind of similar to what I do. I have multiple pumps set up, so you want sort of the water to be oscillating like that, not just going round in a constant unidirectional sort of flow pattern. So schedules that change intensity and direction on timescales of seconds to minutes, and it more closely mimics natural conditions than long unidirectional runs. So our goal shouldn't be to maximize the amount of flow, rather creating an environment that continuously renews that boundary layer without tearing the tissue off. These studies provide fundamental scientifically backed information for SPS-dominated systems. Ideally, we should hold nitrate in the low 0 to 10 ppm range and phosphorate roughly within the 0.0s of a part per million. And we should be maintaining these values steadily rather than oscillating between extremes. Additionally, we should increase light in small steps only when nutrients and flow support the change. Build flow patterns that are alternating and turbulent rather than fixed and laminar, and confirm trends with measurements that are precise enough to detect change even at the low end of the spectrum. It is easier to keep SPS in good condition by maintaining the stability than by reacting to fluctuations with large corrections. A good analogy I like to use is keeping a marine system is kind of like trying to steer a large cargo ship. You're not ever going to be able to steer it very, very quickly. You've got to kind of anticipate where you want that ship to go and make little adjustments on the way to keep it nice and stable. So targeted feeding of small particulate foods can also increase nitrogen and phosphorus differently depending on the type of food you have and the bacterial loading within the tank. If nitrate rises while phosphate remains undetectable, reduce phosphate removal and or add a very small or steady phosphate dose. If phosphate rises while nitrate remains 0, increase nitrogen availability with equally small steady additions or adjust export strategies that are selectively removing the nitrogen. Avoid the cycle of heavy feeding followed by aggressive media changes, as that cycle drives instability and pushes corals through repeated phases of surplus and deficit that they basically can't accommodate. Our lighting plans should also acknowledge that nutrient context changes their tolerance as well. So a colony adapted to moderate PAR under lean nutrients cannot be pushed to high PAR overnight without consequences. Instead, you should try and raise intensity by small increments and hold each change for 2 to 3 weeks while you evaluate tissue texture, polyp behavior, and overall growth. Watch for subtle indications of stress, such as persistent tip retraction, diminished fluorescence, and/or changes in algal density or colors. If these signs appear after a light increase, evaluate nutrients and flow before touching anything else. Re-establish balance and allow the colony to stabilize before the next change. Additionally, flow should be measured at the coral, not in front of the wave makers. Turnover metrics measured at the pump do not describe conditions at the branches, and simple observational tests are adequate. One way I like to try and do it is when I'm feeding the fish, you'll often put little mysis shrimp in there and you can kind of guesstimate how far the mysis travels in the water when it's in there. So I dump a whole lot of food into the tank, sort of set out like a rough sort of measurement. You count how long it takes for a piece of mysis to go flying by, and then from that you can get sort of a rough indication of your flow rates. Any sort of aquarist who's kept corals before or knows anything sort of about flow rates, it's pretty easy to kind of tell what your flow rates are. But for anyone who hasn't done that, that's another easy way. So another thing to check is that if this food or any other particles hang in eddies around branches, the boundary layer is not being renewed. If tissue flaps continuously in one direction, flow is excessive and unidirectional. Adjust pump angles and schedules to produce alternating movement through the colony rather than across only one face of it. Short pulses and periodic reversals create shear that strips the boundary layer without damaging the tissue. In addition, alkalinity and pH influence calcification rates and can also set the energy cost of building new skeleton. A mid-range alkalinity of 8 to 9 DKH with stable balanced nutrients and matched flow and light is optimal, and more representative of natural systems as well. Corals build capacity to grow over weeks and months, so provide the conditions required and allow them to use the energy efficiently. So when growth stops or colour shifts unexpectedly, work through a short diagnostic sequence grounded in the mechanisms we've just discussed. Confirm that both nitrate and phosphate are present and that neither is being driven to zero while the other remains detectable. Verify that light has not been increased recently without compensating change in flow, and inspect pump schedules for long unidirectional runs and adjust to introduce alternation and turbulence. Resist the impulse to add supplements or replace equipment before these fundamentals are corrected. In most cases, restoring balance and stability across nutrients, light and flow resolves the problems without any further intervention. However, species differences also need to be taken into consideration. Branching acropora with thin tissues and high surface area are quick to reflect the state of the light, nutrients and flow. However, species differences also need to be taken into consideration. Branching acropora with thin tissues and high surface area are pretty quick to reflect the state of light, nutrients and flow, whilst massive species respond more slowly and sometimes appear indifferent until a threshold is crossed, at which point they change quite rapidly. The work by Han et al. Showed that Lobactis scuteria responded less than 2 branching species under the same conditions, which is consistent with field observations and with our own experience. We all know that lobos are probably a bit easier to keep than acropora and other things like that. The 2024 flow study also found genetic level differences within species. Therefore, it is also important to remember that different corals inhabit different areas of reefs, and therefore they may require slightly different parameters, which sometimes can be impossible given the nature of keeping aquariums in these systems. So plan for that diversity and avoid forcing a uniform environment on colonies that need different placements and conditions. So another thing is that dosing is actually a lot easier than most people believe it to be. I get a lot of questions about dosing and how hard it is. It's literally you just set up your doser and you just follow the instructions on whatever you're dosing, and you sort of just keep measuring it over time. But creating or buying dosing solutions and delivering them through a peristaltic pump on a daily schedule smooths out that variation quite a lot. Small daily additions of nitrate and phosphate can reduce the need to use large corrective doses that overshoot. If bacterial carbon sources are part of the export strategy, be aware that the stoichiometry and likelihood of selectively removing one nutrient. Refugia remove nitrogen and phosphorus, but also respond to lighting and iron availability. A change in a refugium photoperiod can alter both nutrient and trace elemental trends in the system, even within like a short period of days. So as touched on before, temperature interacts with flow, nutrients and lighting. Systems operating near the upper end of the acceptable range leave less room for error. The Han et al. Study justifies making consecutive choices in anticipation of heat rather than reacting afterwards. Additionally, your system maintenance and schedules should match your reef tank goals. If the objective is rapid SBS growth under strong light, measure nitrate and phosphate weekly at a minimum, and cross-check methods regularly with reference solutions. If handheld instruments are used, maintain them, replace reagents on schedule, and log results to evaluate trends rather than single points. Make one change at a time and watch the trend. Corals respond to the direction and amplitude of change as much to the absolute value. So basically, if you've got 0 to no nutrients in your system, and whilst it might be more optimal to have 10 ppm of nitrate or whatever, it's going to be far worse off for the corals going from that 0 to 10 ppm than it will be to just keep them at that 0. So try and do it over time, basically. So to make reef keeping easier, I recommend keeping methods simple and try not to force your tank into being something that it isn't. It is easier to keep a system stable with 2 methods of export rather than 4, and it is easier to interpret results when the path from cause to effect is short. For example, like my tanks, I only use 2 methods of nutrient removal, or 3 if you count water changes, but I don't do them very regularly. So on both tanks, I run a skimmer and I also run mechanical filtration. That is basically the only nutrient export I run in these tanks. I know a lot of people have really good results with like refugiums and nutrient removal, dosing and stuff like that. But at the end of the day, it just adds a little bit more complexity into it. And if you have the time to do it, by no means do it. But I like to try and keep things simple. So that's just why I use those. And they're a lot cheaper too. So the use of media and nutrient dosing schemes can solve some scenarios and bring a tank back on course, but they add more moving parts and increase the chance of driving one nutrient to 0 while the other remains measurable essentially. So areas of high detritus created by uneven flow can elevate nutrients near rock, even when the water column tests look acceptable. Where export has to be increased, do it slowly and watch both nitrate and phosphate rather than one only. I think a lot of people see these stunning clean systems online and try and set up a tank which mimics these. However, I often tell people the dirtier the tank, the better. Whilst my glass and sand might look immaculate, my sump is absolutely filled with detritus, algae and bacteria, which serves one of the most important roles in the entire tank. Like, it acts as a nutrient export in and of its own. We expect to have these sterile systems, which is not representative at all of how natural systems work. Anyone who's ever been out on a reef will see that under all this beautiful coral are just layers and layers of algae and bacteria and everything like that. And they form your microbiome forms just as much an important role of your reef tank as your maintenance schedule or habits. So taken together, these 4 studies establish some credible information for our hobby. Balanced low nutrients support growth and normal tissue condition over months. Trace balanced additions delay bleaching and maintain calcification under heat and high light in multiple species. Long-term low-level enrichment builds tissue and symbiont reserves and improves performance under stress. Moderate dynamic flow maximizes growth and survival, whereas stagnation, excessive unidirectional flow reduces both. For SBS-heavy displays, parameters that reflect these findings include stable nitrate between 1 to 5 ppm, phosphate between 0.03 and 0.08 ppm, carefully acclimated par matched to the coral placement, and alternating turbulent flow at the branches rather than high turnover measured at the glass or pump. If the system drifts, try and change one variable only at a time and hold long enough to see the biological response. Expect corrections to play out over weeks rather than days. For mixed reefs, maintain the same balance but with slightly broader tolerances. The large polyp corals are often more accepting to higher nutrients but will still react poorly to skewed ratios. Soft corals can mask instability by continuing to grow while SPS retract. So rely on measurements to detect trends. Place species with similar needs together so that flow and light can be tuned by zone rather than forcing one setting across the entire tank. When a colony pales at the tips while bases remain coloured, look first at recent light changes and flow. When colour deepens while growth edges stop, evaluate whether phosphate has been driven too low relative to nitrate. When a stable system begins to show day-to-day swings, check physical causes such as clogged mechanical filtration, refugium lighting, timers or pump schedule overlaps that create long unidirectional runs. The biology is often responding to physical changes that went unnoticed. Additionally, timescale matters, so the 15-month enrichment work makes it clear that resilience is built slowly. If you're correcting a lean highlight system, add both nitrate and phosphate in tiny daily doses, hold the new baseline for weeks, and resist the impulse to tweak daily. If you're correcting imbalance, reduce the removal method that is driving one nutrient down, provide a minimal daily dose of the missing nutrient, and wait for that trend to stabilize before taking the next step. These topics I discussed form the foundation of our hobby. However, everyone's tanks are different, so it's important to find a method that works for you and your tank. However, everyone should aim to maintain measurable balanced nitrogen and phosphorus, and hold a conservative lighting plan and raise intensity only when both nutrients and flow support it. build a flow schedule that is alternating and turbulent at the coral, check the system for ratio drift on a regular basis as well. So thank you all for watching. I hope you enjoyed this video and I hope you all learnt something. I certainly did. I like reading these papers and I find it good even just talking to you guys and going through it and creating these videos just to learn more. So if you have any sort of ideas on what you'd like to hear about next, please leave a comment below. And if you like these series of videos, I'll probably do some more of them. So yeah, thanks for watching.